All right, about a minute early, but I'm going to get this thing started. Welcome, everyone. Greetings. For those of you who don't know, my name's Ian Taylor, and I'm a director here at FlowJo. Today, I'm going to be talking to you about plugins in both SeqGeek, our latest software for single-cell RNA sequencing analysis, and in FlowJo as well. And this slide deck is a little bit thin, but that way I can get to the live demos all the quicker. While I'm going through this presentation, if you have any questions or concerns, feel free to just post them into the question and answer section, or just mark them in the chat and I should be able to see them while I'm going through this presentation. So, what are we going to be talking about today? Again, this is all about plugins. So I just want to start by pointing out that we host all of the plugins available for both SeqGeek and FlowJo. FlowJo.com. Documentation available, some of which describes the plugins at docs.com, either /D2 or in the /SeqGeek documentation. Another great resource that you can visit if you need more training is FlowJo University. The link there is available. You can also write to tech support, which is just techsupport@flowjo.com, or SeqGeek@flowjo.com. And my personal email is taylor@flowjo.com if you have questions for me after the session. Again, just a brief overview of what we're going to be talking about. Just getting started in plugins, how to essentially connect the plugins to FlowJo and then FlowJo to R, given that some of the plugins do have R dependencies. And then I'm just going to briefly go over a few examples in the slide deck, and we can actually go through them live in the software afterwards. Two in FlowJo are Calibration and Teast. We also have Seurat and ST-Impute. It looks like I didn't add FlowSOM, but we will briefly be talking FlowSOM as well. That's a plugin for FlowJo. And also most recent plugins, those products. We'll also talk a little bit about troubleshooting. In case any of you out there are interested in creating your own R, and you've already created really cool R scripts, you can create your own plugins in Java based on those R scripts as well. And then again, the fun part, we'll do a little bit of a live demo, see if we can't run some of the plugins using the softwares today. So without further ado, let's talk about getting plugins set up in FlowJo. First of all, if the plugin doesn't come with FlowJo, and we do provide a few plugins with the software when you first install, you'll have to visit exchange.flowjo.com in order to download the jar files associated with that plugin. If the plugins have R dependencies, you'll have to install R, and you can reach that through this link here. Configure your dependencies in FlowJo. This simply means follow the instructions on the right. You're going to open up the preferences, and that's done through the little red heart icon in the top right-hand corner of FlowJo's workspace. And then you're going to click Diagnostics. And from there, you're going to be able to set up your R path. On a Windows machine, if you don't know your R path, you can just search your program files folder for R and find the correct bitness of your R executable and just point FlowJo there. On a Mac, it's a little simpler. You can open up your terminal and type in the command which R, and it'll return something similar to what I've shown here in the far user local bin and then R, and you'll just paste that in to your path. You'll also need to have a directory plugins. Now, it's a little different on Windows and Mac, but in my case, I like to place a plugins folder on my desktop. I'll show that to you once we're in the live demo as well. You'll also need to install any packages in R, which these plugins depend on. And if you follow the instructions when you first download one of the plugins that depends on R, it'll give you instructions on how to perform those installs. And it's usually a pretty simple process. So again, just to go over some of the examples I want to touch on, Calibration is a new and pretty popular plugin that allows you to calibrate parameters if you have something like beads, MESF beads that tell you exactly how many molecules of a given fluor are on your beads. Then you can utilize that to calibrate parameters within your data. You can also, well, yeah, we'll get into that a little bit more. TSNE, which is a really popular plugin for FlowJo, not quite as new, but again, very popular. It allows you to do T-distributed stochastic neighbor embedded mapping. So this is a way of basically getting clusters out of data for the purposes of discovery, typically. If you have many, many parameters, for example, and you can't look through them one at a time, TSNE can help you tease out the variable populations that you would want to gate on. FlowSOM is another clustering algorithm that's developed outside of FlowJo, but we've packaged up that R script in a way that FlowJo can reach out and run the FlowSOM package, returning some clusters to you and doing minimum spanning trees, similar to what Spade would produce, but we probably recommend FlowSOM before running Spade. We also have quite a few plugins coming to SeatGeek, and two of the newest are Seurat, which is a plugin that references a pipeline developed at the Satija lab. It's a really popular way of basically performing a whole pipeline of analysis in R for single-cell RNA sequencing data. And so we'll hopefully be able to walk through a little example of that. And finally, we've just released a new plugin, SC for single-cell Impute, which does it calculates dropout events in again, sequencing data. And it's really good if you want to go ahead and perform gene set enrichment, then you'll probably want to correct for dropout events ahead of time. So that's a really handy plugin. All right, so just referring to calibration, I've thrown some screenshots up here for what you'll see if you run the calibration platform. Now, our calibration platform has no dependencies in R, so you don't need to worry about connecting to R if you're going to be running just the calibration plugin. When you first fire it up, it's going to add which parameter you want to calibrate. And in this case, I've chosen FL1. It's then going to walk you through some steps. And if you choose auto peak, it's going to do some peak finding on the data within the parameter that you've selected. And it's going to feed you back the mean fluorescent values for that channel that you're calibrating in the calibration data set. And here we've just entered in a few numbers as if we're going to calibrate based on a known set of molecular equivalents. And once you've finished and calculated your calibration, it's going to provide you with develop value so you know how well that regression fits the actual data. And this is a linear regression. You're also going to get a set of peaks that are the peaks auto-detected for your data in the workspace, and most importantly, a derived parameter, which is the calibrated parameter that you can apply then to any other appropriate data files that contain the same parameters that you've been calibrating for. So that's been really handy. We've seen a lot of really good user feedback on that plugin. Next, I want to talk a little bit about TSNE. For all of our plugins, in order to run the plugin, you're going to have to select a population. And so in the case of TSNE, it's no different. You're going to select your population of interest. In this case, it looks like we've chosen a subset that we downsampled. And after selecting that population, you're going to go to the Workspace tab in FlowJo's Workspace and look for the plugin section there. You can see it in the middle dialog. If you click on that, you'll get a list of all plugins currently connected to FlowJo, and then you'll choose TSNE. What you'll get is the TSNE platform, which appears on the right here, or sorry, on the left. And it's going to automatically try to select your compensated channels. Now, if you've done gating upstream of your TSNE mapping, you may want to disclude any parameters already taken into account in your gating strategy. In this case, we're just going to use all compensated parameters. There are a few different settings that you can adjust. If you move the iterations all the way up to 1,000, it's going to take a little bit longer, and this is a computationally intensive plugin, so this can take quite a while depending on the number of events, the iteration, the perplexity, all the different kind of settings that you can tweak. That's one reason why you might downsample a population before running TSNE, is just to speed up the mapping that occurs. Finally, once it's completed its calculation, you are going to get two new drive parameters, tsne(x) and tsne(y), which are going to try and describe the variance in your data through a 2D mapping. So it's going to illustrate all of these parameters that you've calculated tsne on in two-dimensional space, trying to preserve the differences between populations. And what you see on the right are a set of tsne plots. In the topmost plot, we're just looking at a pseudo-color version of this. And in the bottom plot there, you're going to see a tsne where we've done color mapping and utilized CD3. So those populations that are high for the CD3 marker are going to show up in red. And those events that are sort of in the mid-range of CD3, looks like don't have a lot of very, very low events, but the middle events are showing up in green. So those are maybe CD3 dim, and we're probably going to exclude them from our T-cell gate. The next plugin I just want to briefly touch on before we get into demo is the FlowSOM plugin, which again is one of the most recent releases for our FlowJo platform. This is going to do some clustering similar to what Spade does, except I'd like to think a little bit better. Here on the left, we see we're basically just selecting a population on which to run the plugin. And then in the middle there, we see the platform, the FlowSOM platform, where you're going to select the parameters on which you want to perform this clustering and set some of there are a variety of settings here for FlowSOM that you can adjust, including creating a PDF and a minimum spanning tree. And in this case, I've just used the default settings. And on the right, we see the minimum spanning tree that's been created. You'll notice there are little pie charts for each of these clusters that have been developed. And the pie charts correspond with a key in the top left, which illustrates basically how much any given parameter is contributing to this cluster. So that's a pretty cool new feature that we've implemented. I believe this was developed by a group of Belgian researchers. Maybe University of Ghent, I want to say. Okay, so that's the plugins that I want to kind of touch on for FlowJo. If there's any questions, I would invite you to toss them up on the Q&A board. Otherwise, I'm going to move on to some plugins in SeatGeek. And again, just a brief overview before we actually demo on some live data. All right. So one of my favorite plugins is this. Oh, sorry, there were some questions and I didn't. Can I compare groups of samples by tsne? Absolutely. So if you're going to be comparing, let me back up here a moment. If you're going to be comparing groups of samples in tsne space, you're almost certainly going to want to combine those samples ahead of time. Something I didn't touch on, the tsne algorithm is non-deterministic, meaning if you run it on the same population twice, even with the same exact settings, you're going to get a different mapping. That's because there's a random seed involved in the algorithm. But if you combine your data files ahead of time, then after you've run tsne, you'll be looking at all of the samples in the same tsne space. And there's actually ways you can tease apart the different samples that were combined downstream. So just a brief overview, if you were going to try and do this, you would load the samples of interest into a FlowJo workspace, probably gate down to your populations of interest, select those populations all at once in a given workspace, right-click on one of the populations that's been selected and choose Export Concatenate. Once you've opened up that Export Concatenate window, make sure you are in the Concatenation tab. And when you concatenate the files together, you'll get one sample that comprises all of the events in whatever populations you had selected before. When you run tsne on that concatenated file, again, you'll have a common set of tsne parameters with which you can look at it and compare between different samples. So that's the way that you would do that. I know this is a little bit of a complicated process and it's definitely multi-step. So feel free to write to either myself or to techsupport@flowjo.com and we can absolutely walk you through this process. It's very common. Lots of people are running tsne and so we get this question all the time. That's a really good question. All right. That being said, let's see, we're on to Seurat. So as I had described a little bit earlier, Seurat is a pipeline developed by the Satija lab. which is a really popular group from NYU doing some really cool stuff with sequencing. And this Seurat plugin, named after the famous pointillist, French pointillist artist, is designed to walk you through a whole set of, a whole pipeline of analysis. And so it'll actually start with a raw sample or a population, and when selected and run, it will do some quality control based on gene quality and cell quality. It will then run a normalization on the data matrix, giving you a normalized matrix. All of this is done basically in one step, which you can see in this window, all the different settings that you would choose. It'll allow you to choose a set of genes of interest or gene set that you want to have these results for. And then once you click OK, it is going to be, again, a computationally intensive process, so it'll take a little while. But the results are kind of amazing. It will run PCA-guided t-SNE, so it'll do the t-SNE mapping for you. And then it will run graph-based clustering using a k-nearest neighbors algorithm on that t-SNE map to give you clusters in both genes and in cells for the data that you've selected. And what we can see on the right here is for sample 2 in a given data set, calculated Seurat clusters in t-SNE space in the image on the right. This was produced through just this one platform, and it's something that you would probably want to validate and verify after the fact, but oftentimes you'll find that the Seurat is using these machine learning techniques to find some really interesting populations and pulling out the actual biology that you're potentially looking for. As I mentioned, it comes up with a set of clusters, which could be the cell types within your data set or pathways or states that are being played out in the biology. You can also get, it will also provide you with gene sets, which represent the most different genes within each of those clusters in the gene set area of SeqGeek. All right. So that's Seurat in a nutshell. The other one I want to touch on, mostly because it's the newest plugin that we've released, and also because it's very powerful, is the SC or single cell impute plugin, which imputes or infers dropout events from an expression matrix and is very useful for gene set enrichment analysis, where you want to make sure that dropout events are correctly accounted for. And basically, it's going to move values that were 0 into into an expressed state based on a set of inferences. Slightly computationally intensive, so if you're doing this for thousands of cells, you may have a little bit of wait time. But the way it works is essentially once you run the plugin, you'll get the dialog on the left. You'll select the genes that you want to run this for. If you have any categorical parameters in your data, you probably don't want to include them. You're going to have to guess the number of clusters that you're dealing with in the data set, which you may be able to make an educated guess on based on, upstream PCA or even t-SNE mapping. And then once you have that set, it's going to calculate for a little while. But once it shows done in the plugin, as you'll see on the image on the right, it is going to import a new data file, which has the suffix cimpute.csv. And that is a new file that has had the impute process completed on it. So you'll have a new data file that contains what we might call like a normalized expression matrix or an adjusted expression matrix. And those are kind of the plugins I want to cover. And like I say, I want to get into actually playing with them in the software. But before I do, I'll just talk about some tips and tricks. So again, as this user was asking previously, or watcher, I don't know if you've downloaded SeqGeek or FlowJo yet, but if you're going to be running something like t-SNE, almost certainly want to concatenate all of your populations of interest together so that everything can be seen in the same t-SNE space. And you want to be sure to configure R correctly. Now, this can mean downloading an older version of R or updating your version of R. This can include installing R packages on which certain plugins will depend. For example, there's a whole Seurat package, and it's rather large. If you're going to be running Seurat, you definitely need to have that installed in R. So those are all considerations. If you are, as I mentioned previously, a developer and you write your own R scripts, like maybe you're a bioinformatician, you have a great tool that you'd like to give researchers the ability to run on their own, you can actually package up, or we can help you package up R scripts into jar files, which can then be distributed so that researchers are able to utilize the tools that you've built. So that's really handy. If you wanted to try and do this yourself, we highly recommend the following developer guide or documentation, which describes in detail how to create a plugin for FlowJo. Again, these are plugins that reach out to R, although you could actually, in theory, build plugins that reach out to Python or other external third-party softwares. And we've done quite a bit of this ourselves in tech support. So if you have questions there, and I anticipate you will, feel free to write to techsupport@flowjo.com. Even if you're not interested in packaging your tool, maybe, if your tool seems cool enough, we would be happy to help you out with that as well. Before we move on, I'd like to see if there's any more questions at this time. Otherwise, I'll jump right into it. And I don't have any questions, but you want to reach out later, feel free to grab these email addresses for either myself or the technical support team here, which is we get back within 1 business day, sort of as a hard and fast rule. So do feel free to reach out if you have any questions on plugins or anything else with regard to our platforms. And with that, I'm going to leave this help information open while I go through some of these demos in case you want to take down notes on that. Maybe we'll start with SeatGeek. As you can see, this is just a blank SeatGeek workspace. If you haven't tried SeatGeek out yet, I would highly recommend you go to the FlowJo.com website and download, register for a trial of SeatGeek, and you'll be able to use it for 30 days free. And again, if you have questions, please do reach out in that regard. First thing I'm going to want to do here is just add data, and this works very similar to how it would in FlowJo. We could drag and drop a data file in, but I'm going to load it from a folder where I know there's some cool data. This is 6,000 PBMCs, so peripheral blood mononuclear cells, and it's actually 5 different samples combined, which gives me a really great chance to use our auto cat gate or auto gate for categorical parameters plugin. If you go to the workspace tab, you'll see there's a plugins section, and there I've got a bunch of plugins here. The one I want to start with is auto gate categorical, just so I can separate out my different samples. And the way this plugin works is basically you're going to scroll through your parameters, in this case many genes, and we're going to find this sample ID parameter. If you didn't know, like in this case, categoricals all appear at the bottom of the list. If you didn't know where that was, imagine this is just somewhere in the middle of our thousands of genes, I could just type in sample, and I get the sample. It pops right up there, and if I click OK, it's going to run this auto categorical parameter plugin, which does some peak finding and tries to figure out which are my discrete populations within a categorical parameter. So this is perfect if you have like a treatment, anything that's been separated out ahead of time and you've entered it in as a parameter to your data matrix. The auto categorical plugin should be able to separate them out. And just for demo purposes, I'm going to utilize the smallest of our populations there in order to go ahead and run another plugin. Maybe we'll start with Seurat, since that may take a little while to finish calculating. After you've clicked on that plugin, you'll notice you get this screen that we had described. I'm going to leave everything mostly default. Actually, I might back out of this momentarily. I'll show off another feature in SeatGeek, even though it's not a plugin. If we go to the discovery band, in our latest, select this first. Go to the discovery band. There's now a quality control button. As of just a couple of days ago, we've included this. And with this, I can pick out some genes of interest. Let's go to our gene view. So in this plot, what I'm seeing are a set of dots, each representing a gene, as opposed to a cell that we might be used to from FlowJo. And here I'm going to gate just sort of almost a singlets gate, starting at a value of 10. Moving all the way up here, I'll leave out those last few highly expressed genes, and we'll call this genes of interest. This gives me something to run Seurat on, so I'm not running on the entire data matrix, which again may take quite a while. And you'll notice that the gate I've drawn actually creates a gene set here in the gene set area of the SeatGeek workspace, which is pretty cool. We could actually export this or take a look at all the parameters that are included there, but I'm going to skip over that because we're working on plugins. So back to Workspace tab, and if I choose Seurat, we'll get that same dialog. Then I'm going to leave everything default, but I'm going to choose my genes of interest on which to run this. Can't scroll because it's too much stuff. Notice it included sample ID. That parameter we want to exclude because it's not a part of our expression matrix, strictly speaking. And I'll use the negative binomial normalization. Okay, so while that's calculating, I'll close these windows down and maybe focus on the other plugin that I demonstrated, which is our single cell impute plugin. We'll run it on sample 3 here. Now again, if I go to the Workspace tab under plugins, you'll see single cell impute has been installed on this machine and connected to SeatGeek. If I choose, again, let's take a small subset of the genes, half of these, and maybe reduce the number of clusters to 5. And it even warns me it could take 2 hours. So let's not cross our fingers that this is, we'll cross our fingers that this is going to work, but we won't hold our breath. How about that? So these are just a couple of plugins now calculating. While that's happening, they're actually being done on different threads. So running single cell impute is not actually slowing down my Seurat calculations. There may be an argument, there's an argument to be made that I could run single cell impute before doing Seurat, just so that I'm dealing with an expression matrix in which dropout events have been corrected for. But meanwhile, I might just move over to FlowJo and show you some of the plugins that are available here. So to start with, again, I'm just going to load some data. And in this case, we've got tens of thousands of cells in each of these samples, and this would be kind of a typical run. Oh, looks like something finished. So the impute script failed. And it's good that you get a little warning here. It's unfortunate that we didn't calculate that extra sample, but it gives you at least an idea how that would work. Within the Seurat plugin, however, we have calculated p-values associated with each of our genes in every single cluster that was developed. And I'm not sure yet how many clusters we have. If we go over to the workspace, it looks like there were 4 clusters generated, each of which should have a set of DE genes. And it looks like, so for an example, our first gene set, DE genes for cluster 0, contains 116 parameters. And those are all illustrated here with associated p-values indicating significance or reproducibility within the experiment. And most of those look like they're pretty significant. So, I mean, by that I mean very small numbers. The largest of which is 0.0097. In case you're curious. Once that's calculated, it's actually got a PNG image also along with it, which can be added to the layout editor simply by dragging the plugin into an open layout like so. And these are, of course, violin plots, which show us some of the coverage that our genes provided for each of the clusters. So we can see whether or not there are certain genes that are particularly splitting the data. So is it bimodal? We can also see whether or not that there's a large spread of data, or whether or not it's kind of clumped at the bottom, which would be a sign of poor quality. And that's all well and good. But I think what's most impressive after having run the Seurat plugin is to take a look at the TC mapping and clustering that's been performed. So here I'm opening up the graph window within SeatGeek, which shows us all of the gene sets and the gene parameters that are contained in this data file. But what I actually want to look at are not genes, but t-SNE parameters. So let's just type that into search. There's our t-SNE X. There's tsne-y, and already I can see that there are some nice clusters there. In case that's hard to see, we could change this from pseudo color to something like density. Density is not a good choice. How about contour? That's a little better. So we can see that there are some nice islands being grouped together here in this tsne mapping. So that's telling me that Seurat did a great job at actually setting those PCA guided tsne parameters. But then what if I want to know where these clusters lie? Well, Seurat creates this handy cluster parameter, categorical parameter, that allows us to separate out all of the different clusters that it developed. So in case I'm going too fast, basically you're just going to click this color axis checkbox, click on this drop down and choose gene sets and genes. From there we get a list again of all of the parameters available. And the one I know I want to look at is cluster Seurat. So if I type in part of Seurat, we get that. In there we see very, very nicely that the very nice clusters that have been developed automatically plug in. So this is a really great way to start doing discovery on a data set that you may have, very little knowledge about a priori. Because oftentimes these sequencing analyses can be basically like boil the ocean. We're just looking at old transcripts, and out of here we have 26,000 parameters, more than 6,000 parameters. Good luck being able to identify a population like this without some sort of assistance, some machine learning or clustering tools, which SeqGeek handily. Hope you all are able. I've lost here. They've gone down. Thanks for bearing with me. If you're having trouble hearing me or seeing me, or if things are breaking out, feel free to just drop something into the chat or question area, and I'll try and adjust. But hopefully we're back in business. So back over to FlowJo. In FlowJo, there's also a set of plugins. And to start with, I might just downsample one of these populations to show you how we can reduce the number of events that are seen for a given sample. And it's going to ask me to save my workspace, which is no problem. Go ahead. Just use the default there. And we want to go down to 5,000 events. We'll call this 5K. Oh, I guess I can't add numbers there. How about this? 5K. And notice we got a population immediately calculated from that plugin. So downsample plugin just allows you to select an evenly distributed set of events from your data file. Prior to having this plugin, we used to have people gate on some unbiased parameter, say event number or time, because normally your collection isn't biased in terms of the time of collection of the events. But the downsample actually helps in this case because it assures that there is no bias. Even if somehow you were collecting maybe lighter events first and heavier cells later, you're going to get an even spread of that data if you use the downsample plugin. Now that we've downsampled, this is a really convenient time to take a look at the tsne or T-distributed stochastic neighbor embedding plugin in FlowJo by clicking plugins and choosing tsne there. Notice no parameters appear to be selected, but it has found all of the compensated parameters, which is typically what you would want to use when running a tsne calculation. Again, this is computationally intensive, especially running at high iterations. So I'm just going to go ahead and let this run. I'm not going to generate the movie. That's just going to slow us down. And we'll kind of just let this run in the background while I show off. Maybe I'll be daring enough to go with the FlowSOM plugin. And again, this is going to do a sort of clustering on the data. I'll use the compensated data this time as well. And I think I'll just go with the defaults as far as settings. If you ever had questions about what these different settings do, oftentimes we'd recommend just mouse over the setting and you'll get a little tooltip and it'll describe what any of these fields is going to do when you make adjustments there. There's also documentation that comes with the plugin when you download it from the Exchange, so that's another great resource, as well as we try and list all of our contributors here at the bottom. So if you wanted to look up any publications they have, they're available as well. And once I click run, it's going to begin calculating those FlowSOM clusters. It's also going to generate that minimum spanning tree PDF. All right, so that was actually very quick. I noticed our tsni is finished as well. But let's just take a look at the FlowSOM now that we have it. If we open up the layout editor, we can see this FlowSOM image just by, again, all I did there was drag this plugin node into the layout editor, and we get a minimum spanning tree from the FlowSOM plugin, which is pretty cool. So I'm not gonna, I guess I'll just touch on this key here, which describes basically the pie chart that you'll see for each of the clusters, in which a different color in the pie chart represents the contribution of a given parameter to that cluster. Each of the different colors here, sort of the haze around that pie chart, represents the meta cluster that was developed and placed as a gate back here in the, I'm looking at SeatGeek now, back in the FlowJo workspace. So I've got populations 0 through 7, so 8 populations. Each of those is going to correspond with a different color here. And again, there is a legend that kind of gives you a description. So we can see that, for example, looks like population 6 is our purple set of meta clusters. You could also map this with tsni if you wanted to. Basically, if we had run tsni on sample B2 here, we could do like an overlay to see where each of these clusters falls within a tsni mapping and see if the tsni would pull them out as well. Just to show the results of the tsni plugin here on our 5K population, we developed through downsampling. I'll open up a graph window. Notice you have these A over B derived parameters for our downsample, as well as tsni X and tsni Y. So that's telling us that there are a new set of parameters in this data that have been calculated. tsni X, so this, you can already tell there are some peaks here that may be separating out populations of interest, and tsni Y. So that's pretty cool, right? You get all of these different populations just by running this one mapping plugin. And if we wanted to begin investigating those, there's all different ways we could. For example, maybe we're interested in a particular island. I'll use the auto gate to try and grab, based on contour, this guy, and I'll just call it pop1 because it's a bit of a mystery. I'm not sure what it is yet. One way that we might go about trying to figure that out is to open up the layout editor and create an overlay. Call this tsni discovery. Oops. Great. Now, the last set of parameters I looked at were tsni, so I'll just drag this in. And I'll overlay with pop1. I sort of like to see the background black, and then our population of interest in red. We're not too interested in the sample name here because they're all in the same sample. Okay, great. I'll leave that legend just like so. Breaks, make it a little cleaner. We could also show this in like a content. That's nice to see. So what did I do there? I just double clicked on this overlay. Graph definition window pops up. In the specify tab under type, I can change the plot type. So I've chosen contour plot. Now if I click apply, I don't know, it makes it a little clearer. We might want to see outliers. Yeah, it doesn't look like there's a lot of outliers there, does it? That's okay. But now we're curious kind of what is population 1? Well, a really handy tool for that is the n by n multigraph overlay. So if you right click on an overlay, you'll get this drop down menu. If you go to make multigraph overlays, you can choose n by n. And let's bring this down and make it much larger. And what are we seeing here? Well, we're seeing an overlay of population 1 on top of the rest of the 5K data set, the population that we identified through downsampling, and we're seeing that for all other parameters. And so what are we seeing? Well, here's CD4. Looks like POP1 is CD4 low, right? If we look at CD8, so killer T cells, we see that POP1 is very high. So this may be, this may represent our killer T cells. I noticed some parameters here that I'm not particularly interested in, say event number. And if we wanted to remove that, we can just right click. We can see all of the populations that are, or sorry, all of the parameters that are currently being used for this n by n. I'm going to remove our downsample drive parameter, and I'm going to remove the event number. And that leaves us with a really clean, I think, way of starting to investigate which parameters are contributing to population 1. There are other which would allow us to show the contribution of any given set of parameters to a t-SNE population. So I find that's very handy. The other thing we could do before I move on is, especially now that we know which parameter we're looking for, if we color the axis, here's by CD56. So our CD56 high cells might be these green guys, those green islands. But if we set this to CD8, we see that the population we sort of randomly chose is higher in CD8 than maybe some of these other islands. Although we might want to investigate this one as well if we're looking for CD8 in particular. And what else did we mention? CD4 was rather low there. Okay, so maybe these are our helper T cells. We would need to do more gating, obviously, to confirm that, say, checking what CD3 is all about in this case. But if we're talking about T cells, I digress. And with that, I think I've kind of demoed all of the plugins that I was hoping to during this webinar. But if anyone has any questions or wants to see particular plugins used, I would welcome those at this time. So I'll just hang out here for a moment, maybe scroll through some of those other plugins just to remind myself if there was anything else we want to see. Here's a question. CD45RO is not quite homogeneous. What's your opinion? Let's just check that out. So CD5RO, I think maybe this questioner is referring to the color mapping that we're doing. So let's switch to CD45. And I'm thinking this is what you're referring to when you say non-homogeneous, and I totally agree. It looks to me like this t-SNE mapping is not pulling out a strong, basically, correlation between any of these islands with CD45RO. The closest that would be might be this population here, sort of below our POP1. It looks like it's higher in CD45RO than maybe this island over here. But if CD45RO was your parameter of interest, you might have done some gating upstream prior to running t-SNE on the population, so that you've ruled that out as either you're looking only at CD45RO positive or CD5RO negative based on your controls or your standards. So yeah, that's a great question. Oh, yeah, I think this is so. Questioners. Can I statistically analyze differences between the t-SNE populations after having done t-SNE on a concatenated FCS file that I got by concatenating 2 groups of samples? Yeah, so another really great question. You absolutely can, and I would like to demonstrate that for you now. So how would I want to do this? What I'll start by doing is just applying this downsample to the whole set of data. And I probably won't use every single sample, but just to illustrate. What this is going to do now is calculate a random set of 5,000 events for every single sample here. And there's 9 samples. What I'll do is just select maybe the last 4 here once it's finished. Looks like it's taking a little while. Usually it's much quicker. And because it's recalculating that 5K set of events for the previous data, it has to recalculate the t-SNE as well. So if I select, let's see, maybe just hold in Command to select 4 of these guys, I can now concatenate them. And in order to do that, I'm right-clicking on one of these and choosing Export Concatenate. I'll go to the Concatenate tab. And within here, I'll make sure I'm exporting as FCS3, so this will re-import into FlowJo. I know that I'm exporting to my Downloads folder, And I'm including all uncompensated parameters. This is probably the hardest to understand, but basically what's happening is all of these samples share the same acquisition-defined matrix, which is stored as keywords in a data file. So if I export all uncompensated parameters, they're going to remain compensated, and I'm going to have the ability to look at both compensated via those acquisition-defined keywords or uncompensated versions of all my parameters. So if you have an long story short, if you have uncompensated if you have acquisition-defined matrix, exporting all uncompensated parameters is often desirable. So concatenate. Looks like I already have a file of the same name. Yes, go ahead and overwrite. And what I want to do is reopen that in maybe a new workspace. Let's just start fresh. So now cancel that window. And I'm just going to close this workspace because I think we're done here. But in this concatenated file, we have a parameter which separates out our different samples. So that's really handy. If I open up a histogram of sample ID, and there we see these are the 5,000 events from sample 1, sample 2, sample 3, sample 4. And I will go ahead and name them as such, just get them separated. Sample 1 looks like sample 2. Hopefully very soon we'll have an auto cat gate for FlowJo that is coming down, like I say, relatively shortly. But for now, you kind of have to gate manually. And sample 4. Great. So now we have a concatenated file, but we've separated out our samples. So what was the use? Well, what we want to do now is for each of these events, each of these samples, with each event within each sample, we want to calculate tsni parameters. So I can do that again, just workspace, plugin section, go to tsni. It wants to save this workspace. I know we're going to overwrite our previous one. Well, that's fine. Yes, overwrite. Make sure we've selected all the compensated parameters, assuming we haven't done any gating that might bias this, but that looks fine. Make sure you haven't selected sample ID, because then your tsni will be separating out based on sample. We don't want that movie. Maybe I'll up the perplexity just to show what that would do. And now we're calculating. So what are we calculating? We're calculating tsni parameters for this concatenated data file. So what we're going to wind up with is a set of tsni parameters that are in common for all of the samples. And that's getting to your question, because what we can do then is compare what were the tsni maps for each of the samples. Spoiler alert, basically these samples are very similar. They're run in the same plate with the same treatment with the same cells. So we're almost expecting to see this be a very unbiased tsni based on samples. But if you had different treatments, different patients, time courses maybe, the different samples that you've combined may bias the tsni in a way that's desirable. In other words, you might start pulling out populations that differentiate the different samples. And that can be something really useful in the process of discovery, or like I say, if you're doing some kind of a treatment course. All right, get this window out of the way. We're not interested in that anymore. Can I concatenate another question from a user. Can I concatenate two FCS files with a different compensation? The same parameters are measured, obviously, but slight differences in compensation is necessary in some cases. Very fair, very true, especially like if it were a time course. In that case, I would recommend you not export uncompensated parameters, because as you will note, the acquisition-defined matrices in those cases, or matrices calculated in FlowJo in those cases, won't match. So in that case, simply choose export all compensated parameters instead. So it's a pretty simple workaround. The one thing that you'll notice if you export all compensated parameters is the name of the parameters will change. Not for individual samples, but for that concatenated file, the name of your fluorescent parameters will be prefixed with FJComp, which simply indicates that FlowJo has performed the compensation ahead of time. And so, yes, you can do that. Really great question. Glad you asked that. In fact, while we're waiting, I might just illustrate what that would look like. So if we select these samples again, let's right-click, export. Now, if I export as is, it'll export all 4 files, but if I choose concatenate, it should combine them again. And if I choose all compensated parameters as opposed to all uncompensated parameters, if we imagine that each of these samples has a different compensation matrix applied, what FlowJo is going to do is take the compensated versions of those parameters and combine them. So you'll get parameters in the new data file that are in common, but they've been pre-compensated with whatever compensation matrix was applied ahead of time. So if I concatenate these now, oh no, I don't want to overwrite, sorry, let me back up. Close. I'm going to change the name of this to EG, for example, and I'll try again. Great. And I'll open it in the same workspace just so we can view it side by side. Notice, okay, close this. Notice that this new concatenated data file does not appear to contain any compensation matrix. That's because it has been compensated explicitly by FlowJo during the combination process. So if I open this up, we see our normal forward side scatter, no change there. But if we look at the compensated, the fluorescent channels that have been compensated, they each contain this FJ comp prefix. And if nothing else, that's just telling you that these data files were, that these parameters are compensated and that they were compensated by FlowJo before some export process occurred, either a regular export or a concatenation type export. All right, so while we were talking, we got t-SNE-X and t-SNE-Y calculated for these combined samples. And if we wanted to take a look at the differences between the samples for t-SNE space, we may wind up doing some, yeah. So I'll just compare maybe sample 1 and sample 3. t-SNE-X, t-SNE-Y. So we see a nice mapping of t-SNE in sample 1. Sample 3 will have the exact same t-SNE parameters. So they'll be comparable. The islands will have been calculated together in such a way that it's not differences necessarily only between populations in one sample, but differences between the combined samples that contribute to this t-SNE plot. So there's our same t-SNE-X, same t-SNE-Y. And we see some major similarities, but also some differences, right? So for example, I see that this population here is a lot denser, for whatever reason, in sample 1, and much sparser in sample 3. And that might be a really important thing, but we'd have to do some investigation to find out. Similarly, dense population here, very sparse here. But overall, they're very similar. So maybe another thing to do would be, if we open up the layout editor, we can drag in sample 3, and shift click and remove that, so not overlaying twice. And what we see is that, there's a little bit of each sample in each of these islands, probably. And that means that, there wasn't much difference, there wasn't much difference between these samples that we used, which we already kind of knew ahead of time. These were PBMCs from, similar, if not the same patient. In fact, this is our demo data, so if you're ever utilizing the FlowJo demo, you'll see this same panel of data. And again, we're not interested in sample name because they all come from the same sample. And hopefully that helps you to understand a little bit how you could do comparison between these different samples in a t-SNE space. There's a lot more we could go into here, like actually quantitating the differences. But I would probably just ask if you had those kinds of questions, write into techsupport@flowjo.com, and we'd be more than happy to help you out. You can also reach out to me. Again, my name's Taylor, or sorry, my email is taylor@flowjo.com. Any questions you have, we'd be more than happy to cover. Even if you wanted maybe like a personalized screen share, that would be more than welcome. Questions that you guys have. You've been a great audience and your time today, and be sure to catch the next webinar. And if you haven't done so already, try downloading SeatGeek and test that out a little bit. It can be pretty fun. Thanks, guys.